everyone. First of all, I'm very sorry that I couldn't make up all the videos on time. So this time onwards, I have prepared myself mentally in such a way that every week at least 2 videos I will be coming up with. So here I am making the first ever video. Like, you know, I've been getting several requests for this video, actually for this topic. So this is something very interesting. But before we begin with the topic, let me again say sorry to you, because I was really late in coming all the videos. So sorry once again. So we'll start with that definitely. And those who are seeing my video for the first time and those who have not yet subscribed to my channel, so please subscribe to my channel, because like I've given you the promise, this time onwards I will be coming up with several topics. The topics I would be getting, or actually this is one of the topics that I had been getting for several times, just for a request, sir, please make a video on that. So I really appreciate you people for giving me all these requests, and thank you so much for that. So we'll start with the topic here. So for the first time, I have taken a topic which is known as RH incompatibility. Now, in this whole topic, what are the things that we are going to cover? We will see what is the meaning of the RH incompatibility, why it is required, what is it, do we really know to know about it, how it will take place, definitely what are the, you know, like stages, when, how it will be taking place. When, this is something very essential, when the RH incompatibility will be visible in every individual, that also we will see. And most importantly, do we have any prevention for that? If yes, what is the prevention? If yes, when should be the prevention be given? So these are some of the topics that we will be covering in this video. And needless to say, the RH incompatibility is known as your HDN, or the Erythroblastosis Fetalis. By looking at this, if you carefully look at this particular scene here, it is HDN. The full form of the HDN is Haemolytic Disease of the Newborn. First, try to understand that the meaning of the hemolytic, hematology. Yes, you got it right, it's blood. So hemolytic, hemolysis means rupturing of the blood cells. Now, blood cells will be ruptured of who? Of the newborn. So that is why it is known as HDN, or the Haemolytic Disease of the Newborn. Precisely, if you know, we have several types of blood cells. Precisely, the blood of the newborn baby or the fetus itself, specifically the RBC, the erythrocytes, the red blood cells will be destroyed. And based on that, they have got one more name which is known as erythroblastosis, the destruction of the erythrocytes of the fetus. So I hope this whole scenario is clear to you now. This is something very interesting, trust me. And I will make sure that this topic will be understood by you people. This is something very essential for 10th standard ICSE students, as well as like those who are appearing for the NEET and the competitive exams. 11th standard, you have a human physiology, and there you will talk about this, the body fluids and the circulation. There you will have this along with the other aspects of that. So without much ado, we'll start with this, that is the RH incompatibility. Fine. So we will start with the RH incompatibility now. I will be going very slowly so that you people can actually understand about it. Now, before we begin with the RH incompatibility, I know that you must have an idea about the different types of blood cells that we have in our body. You know, we have A, B, and O, and more to that, we have A positive, B positive, A negative, B negative, O positive, O negative, AB positive, AB negative. So somewhere down the line, children, you can actually see that along with the other antigen, that is antigen A. Like suppose if I say I have blood group A, that means on my red blood cells, if this is my red blood cells, I would be having an antigen A, and this antigen on the surface of the red blood cells is representing my blood group. So this is same for the AB. So both the antigen A and B will be present on the surface of the RBC. So suppose if this is my AB blood group, so definitely I will be having both the antigen. So like this, the blood group is actually categorized. Here, if we talk about RH incompatibility, that means apart from all these things, we have one more aspect of presence and absence of positivity and negativity. Based on that, suppose, let me give you an example here. Say, if I have, say my blood group is AB positive, that means if this is my RBC, AB, so definitely I will be having A as an antigen, I would be having B as an antigen, and here is the catch now. If you can see over here, I would also be having one more antigen, and that is known as antigen D. So needless to say, my moral of the story is, if you have antigen D on the surface of your red blood cells, that means you belong to the category of Rh positive. Similarly, if you are AB negative in this case, say you are AB negative here, just for an example, if it is AB negative for you, let me write it here. Needless to say, you would not be having this antigen on the surface of the red blood cells, as simple as that. So that's about the concept of the presence and absence of Rh antigen in very short, because we have to keep it short because we will be focusing on this incompatibility and how that takes place. As you all know that overall worldwide 80 to 85% of the population is Rh positive, and 15%, rather 10 to 15% is actually towards the negative. So definitely by looking at this particular word incompatibility, you must have understood by this time that if there is a transfusion between a positive parent and the negative parent in terms of their presence and absence of the Rh antigen, that time we will see the occurrence of this Rh incompatibility. Fine. But before we get into that, we need to understand one thing, that is, what are the probable options that I am having? Like, what do you mean by probable options? Say, case number 1, if I have mother as positive blood group and father as negative blood group. Case number 2, if I have mother as negative blood group and father as positive blood group. And case number 3, if both the parents are negative. Case number 4, if both the parents are positive. So first thing we need to identify, out of these 4 combinations, which option will be actually required for Rh incompatibility to occur. And if you remember guys, in the beginning I showed you somewhere like HDN, Haemolytic Disease of the Newborn, or the Erythroblastosis Fetalis. So definitely it is going to occur in the fetus or the newborn baby, not their parents, that is for sure. Then why I have written all these options? I need to understand, if this is the condition of my parents, what will be the condition of my children? Will they be positive or will they be negative? If yes, will there be any incompatibility or not? So first that we need to identify. So if this is the case where your mother is positive and father is negative, in this case, this incompatibility will not occur. Why? In due course of time you will come to know. If this is the case, where mother is negative, father is positive, there definitely the problem will occur in their baby. If this is the case, both the negative, both the positive, needless to say, no such condition will occur. I hope this is clear to you. So in this case, option number 3, option number 4, there is no such possibility. So let's remove them out. Now let me tell you something. Our blood, that is the red blood cells, listen carefully, our red blood cells do not contain any antibody for the Rh antigen. You heard it right. For Rh antigen, we do not have any antibody generally. So when it produces, if you remember, we have a specific mechanism in which antibody is generated in response to an antigen. So definitely, if a particular cell is infected by an Rh antigen, in order to respond to that, they will produce the antibody against that antigen. Now just see one thing. If my mother is negative, that means a little bit genetics. Negative, negative, positive here. So it can be homozygous, it can be heterozygous. So if this is my case, a bit of genetics we are doing here. What will be the probability here? All the probabilities will be positive. I hope you are clear with this. Because at the end of the day, for each one of these, I will be ending up with these results. Right? Now definitely this is my dominant copy. So they are going to dominate over the recessive copy. So the ultimately blood group will be positive. But I've told you my father is positive. It does not mean that it will always and always have the homozygous copy. That means it can also have the heterozygous copy. Can I say that? So if this is the case, so what will be my result? Now you see. Now see the result here. For option number 1, it will be this. Option number 2, this. Option number 3, this. Option number 4, this. So ultimately what I am getting? Positive blood group. Positive blood group. But if you carefully observe here, this is my negative blood group and this is my negative blood group. So chances are there. That means positive blood group father and negative blood group mother can have a baby belongs to negative category. And if this is the case, that means negative blood group mother and positive blood group fetus or the baby, let me write it here. If this is the case, Rh incompatibility is bound to happen. So I hope this is clear to you now. Now let's see when it happens and how it happens. Something very interesting coming up for you guys. Suppose this is the uterus and say this is the mother, right? Don't go for the diagram, just try to understand the logic here. So definitely this is my uterus, and needless to say, I would be having baby inside this. So say this is the baby. This is the baby now. Right? And obviously this is a mother. So right? So that's my mother's womb and my mother here. Now condition number 1. And what is the condition? That is my mother is Rh negative and the fetus is Rh positive, and the credit goes to father, of course. So happy Father's Day. So here now see what happens. In the time of pregnancy, as you all know that blood of both will never get in touch with each other. We know that. So there will not be any transplacental migration of the blood from baby to mother and the vice versa. It will not be there. But at the time of delivery, and as you all know, you have seen the picture, several pictures. Easiest example is the Three Idiots, where you have seen the baby is being taken out of the mother's uterus and the baby is connected with the umbilical cord. And if you know, some of you know also, umbilical cord at the one end, it will be attached with the mother's body with the help of the placenta. So at the time of delivery, what happens? The placenta gets torn out. At that time, the blood of the fetus, a few amount of blood of course, few amount of the blood of the fetus can get an entry in the mother's blood. If this is the case, so can you imagine that there will be a situation in which it would be ending up with, suppose mother is having negative, right? So let's put all the negative blood group here. But as I told you, at the time of delivery, since the placenta will get torn out, some positive blood of the fetus will enter. So how that time it will look like? Though the amount is low. But now listen very carefully. I told you, if a blood group is Rh positive, that means that particular individual is carrying antigen D on the surface of the RBC. So antigen is a protein. You all know that. So if this is the condition is occurring, the mother's blood will be triggered. Triggered for what? The blood of the mother will get triggered in response to the antigen of the baby's blood. So what will happen? In response to that, they will start producing the antibody. And I hope you know, whenever there is an agglutination of antigen and antibody, what happens? I hope you know that. So here what will happen, this negative blood group, in response to this antigen which was being carried by the fetal blood on the surface of the RBC, they will start producing some antibody. So, and how the antibody look like? Antibody looks like this. Why? So you can actually imagine they will start producing the antibody like this, like this. But you see something now here. The question is how the antibody will produce. This antigen, the antigen D, will trigger the production of the antibody. Not only that, it will also trigger the production of memory cells. So let me write it here. That means right after the triggering, what are the things that will produce? Number one, the antibody. So can I write that antibody for antigen D? So it will be known as anti-D antibody. Along with that, it will also produce, most importantly, and I hope you can make out the meaning of that by looking at the name itself. That is your memory cells. Needless to say, these memory cells will store in their memory that if this is the scenario happens, this is what I am supposed to do for the future purpose, and that is what exactly will be stored in their memory, and that is how the name have arrived as memory cells. This I am talking about the first pregnancy. Did you get that? If this is the condition, what will happen? Placenta will be torn out, the blood will get in touch with the mother's blood, the blood of the fetus. This will be the condition. Production of the antibody against the antigen D will take place, and definitely these antibody will start destroying the cells whatever has get in by the agglutination. But here it will not create any harm to the baby. You remember the name? Haemolytic disease of the newborn, or Erythroblastosis Fetalis. It will not create any harm for them. Why? Because by the time they have started preparing the antibody, the baby has already taken out of the mother's womb. Means the baby has already been delivered. I hope this whole scenario is clear to you. So once again, this is happening in the first pregnancy, so it will not create any harm to the baby. If you remember the name, Erythroblastosis Fetalis, or Haemolytic disease of the newborn, because by the time the antibody is generated, and by the time the same antibody starts killing all these by the agglutination process, the baby has already taken out. So delivery is done. But you never know that second chance will not occur. You never know that. Means if the same thing occurs in the subsequent pregnancy, that time what will happen? My dear students, let me tell you, that time this will create a huge response in the body. Why? This is your answer. Memory cells. Why? Because in the first pregnancy, whenever this situation happened, they have stored everything. That's why they are named like memory cells, no? Now you will see if the same thing happens in the second pregnancy. I hope for the first pregnancy the case you are actually clear with. Now let's move on to the subsequent pregnancy, and with the same condition, that is the baby is born again with the positive blood group. Needless to say, once again, happy Father's Day, because that credits will go to the genetics of the father, dominant. Thank you. So let's see now. So now let's talk about the second pregnancy. So let me write it here. Second pregnancy. Now we'll see what will happen here. You know what? Though I have told you that at the time of pregnancy, during the pregnancy, the blood of the fetus, the blood of the mother will not get in touch with each other. That's for sure. So you can ask then, sir, how the nutrition will go from mothers to fetus? How the waste will come out from the fetus to mother? Well, we have diffusion as a mechanism. We have diffusion as a mechanism. So no need to worry about that. So let's get here. Suppose this is the same scenario in the second pregnancy. If you remember, by the completion of the first pregnancy, the mother has already developed the antibody against the antigen of the first baby, if you remember that, right? So let's make here, definitely I will be having few of them in the blood of the mother. So that's my antibody. Antibody against the antigen D. And where the antigen D is located? On the surface of the RBC. And what is the function of that? That is what it actually indicates as if a person belongs to positive category or negative category. So if this is a second pregnancy case, you know what, it is actually expected that when the baby will be out of this comfort zone, the baby is supposed to get an immunity from her mother or from his mother. Now the question comes, how the immunity will occur in their body. The antibody from the mother's blood will transfuse to the fetal blood. Antibody, not the blood. That means the antibody which will be located in the plasma of the blood will be fused from mother to the fetus, and that is how they develop the antibody. I mean, they develop the immunity, right? But here, do you expect these things to occur? No. Why? Now you see. If this is the case, definitely at the time of transfusion of the nutrients and all through the blood plasma, this antibody will also go there. And by the time, if that happens, since in the first pregnancy we had memory cells, they will start producing antibody in no time, and the quantity of the antibody will become huge, will become massive. And what will happen with that? As they will start producing antibody very rapidly. Why this time rapid? Why not that time it was rapid? Because that time there was no memory cells. They generated the memory cells. Now why this is rapid? They had already generated the memory cells over them, and that memory cells will help this time to produce more number of antibodies very quickly. So antibodies will be producing very quickly, and the antibodies will start getting inside this body, in the body of the fetus. So first scenario, it will start killing all the erythrocytes. Erythroblastosis Fetalis. I hope you remember that. So now let's see the consequences one after that. So first thing for sure, what the baby will suffer with? Number one, erythroblastosis, the destruction of the RBC Fetalis. Second point, destruction of the RBC, red blood cells. You know, when a red blood cell is destroyed, the hemoglobin will also be destroyed. If the hemoglobin gets destroyed, I hope you know that as a byproduct, they will release bilirubin from the hemoglobin. Means right after the end of the destruction of the hemoglobin, they will start releasing bilirubin as a byproduct, which is to be released out of the body, of course. So if more number of red blood cells are destroying or being destroyed, so needless to say, more quantity of the bilirubin will be generated, as simple as that. Because more RBC or more RBC destruction, more hemoglobin destruction, more production of the bilirubin. So what? If the bilirubin produces more than the benchmark, they have a toxic action against the neurological tissues. So now, my dear students, whenever you get to know about the neurological tissues, the first thing that comes in your mind is the brain. So definitely all the bilirubin will go to the brain, and it will start destroying all the neurological tissues of the brain. So in this case, the brain of the baby will not be developed, it will be damaged. And that condition is known as bilirubin encephalopathy. What do you mean by encephalopathy? The destruction of the brain. Encephalon, diencephalon, mesencephalon. The term is actually related to the brain. So encephalopathy, destruction of the brain. For whom? For the bilirubin. So it is bilirubin encephalopathy. I hope this is clear to you. Third, red blood cells are being destroyed very rapidly. Now what is the function of red blood cells? It helps to carry the oxygen to different tissues, to every corner of the body. So if you have less red blood cells, what will happen? The oxygen capacity of the blood will also become less. So there will be a moment when this particular fetus will be suffering from an anemic condition, right? If the fetus is suffering from anemic condition, the heart of the fetus will be failed to pump out the blood to send adequate amount of oxygen to different corners of the body. So don't you think in this time, or in this course of time, the baby will also suffer from a cardiac failure, right? So let me write it here for you. The baby will also suffer from cardiac failure, right? If you know, we have liver and spleen in the body, definitely. Here in the Erythroblastosis Fetalis, the liver and spleen, they get enlarged. I hope you are aware about the word of acromegaly, right? So similarly, these fetus will also suffer from hepatic megaly or hepatomegaly and splenomegaly. So that will also be one of the cases. So let me write it here. Hepatomegaly and splenomegaly, right? And as a result of which, the life of the fetus will be in danger. Why? Again, the same answer, memory cells, rapid production of the antibody. So, a very fast destruction of the erythrocyte cells. And it's sad to say that in some of the cases it is seen that the death of the fetus occurs in intrauterine condition only. So that's about the RH incompatibility. So when it is danger in the second pregnancy. Why? Because in the first pregnancy, the mother's body was triggered to produce the antibody. So what? The same was stored by the memory cells. So what? If the second pregnancy occurs, the subsequent pregnancy, the memory cells will not think twice before the production of this antibody. They will just start producing this. This will be the results. And that is what we call as the Haemolytic Disease of the Newborn or Erythroblastosis Fetalis. And that occurs due to that incompatibility in the RH group. Now children, you see, I told you that it will take place in which the mother is RH negative and the baby is RH positive, because the antigen which is present on the surface of the baby's RBC surface, it will trigger the mother's blood to produce the antibody, and that is how it generates. But now guys, can you think why the opposite cannot be taken place? Means when the mother will be positive and the baby will be negative, when it will not take place? Just think. I'll give you a hint. If we'll be writing the answers in the comment box below, that's about the RH incompatibility, which results in the Erythroblastosis Fetalis and Haemolytic disease of the newborn. 10th standard students, 11th and 12th standard students, this is one of the most important questions for you guys, and those who are appearing for the NEET, those who are appearing for the AIMS and all the other competitive exams, this is one of the most important questions. So now let's talk about, do we have any prevention of that? Of course yes guys. Just imagine if we would not have any prevention, how those couple will be having baby. It would not become possible for them, right? So definitely we have a prevention for that. And what is the name of the prevention? The name of the prevention here is ROGAM. It is an artificial antibody against the antigen D, not the natural, it's artificial. So what I'll do with that? Suppose before the pregnancy, when the couple is visiting the doctor, the consulting doctor, and they come to know about, yes, the mother is having Rh negative and the father is Rh positive. So they have a potential or a huge potential to have a baby with Rh positive blood group. So due course of time, what will happen? The mother will be given this antibody, or the mother will be administered with this artificial antibody. So what am I doing? I am not allowing the blood of the mother to get triggered. Before that, I am giving some artificial antibody which will definitely agglutinate the antigens of the baby. So what is my ultimate criteria? So what is my ultimate advantage? Definitely, guys, unless and until the mother's blood gets time to get into the scenario in which they will develop the memory cells, in which they will come up with their own antibody, why don't we just stop them? And in order to stop them, I have prepared something which is known as ROGAM, and purposefully I will inject the same in the mother's blood. What will happen after that? Whenever there will be a transplacental transfusion, this ROGAM will destroy all these red blood cells of the fetus. And what advantage the mother will get? The mother's blood or the mother's body will not even get the chance to get triggered. So what is the advantage? If the blood of the mother doesn't get any chance to get triggered, in subsequent pregnancy, the scenario will not be fatal. I hope this is clear to you now. Are you getting my point or not? I am not giving the chance to the mother's blood to get triggered against the antigen of the baby. So what I'm doing? I'm giving some artificial antibody against that antigen. So moral of the story, I am not allowing the memory cells and the antibody against the antigen of the fetus to develop. Before that, I am putting them. Before that, I am administering them. So what they will do? They will simply mop out all the red blood cells like a sponge. They will wipe out everything. So if they wipe out everything, so definitely the mother's blood will not get any chance to get triggered. And in this case, that condition can be absolutely in a fine way avoided. So that's about, guys, the Rh incompatibility and the Haemolytic disease of the newborn, or the HDN, or the Erythroblastosis Fetalis. I hope the whole lecture is clear to you. If you have liked my video, please do not forget to subscribe to my channel, of course. Along with that, hit the like button, and I've asked you a question, so do not forget to answer that in the comment box down below. Once again, if you have enjoyed the video, please share with your friends so that they can also get the help from my video. Hit the like button and do not forget to subscribe to my channel. And please, if you have some topic in your mind, please do not hesitate to tell. Write in the comment box so that I can come up with the videos, those videos according to your choice, as soon as possible. Till then, bye bye, take care, enjoy the video. Thank you.